IB Bio, Molecular Biology Part 2, focuses on the biomolecule group known as proteins. This is the first of two parts on proteins. The essential idea is proteins have a very wide range of functions in living organisms. Protein function will be the subject of Molecular Biology Part 3. We must first understand protein structure. Here's an outline of all the movies available for studying IB Bio molecular biology. Use this slide to reference the part of the topic that you need to review. This movie is focused here. Let's jump in with an IB syllabus statement that you've seen previously. Outline the role of condensation and hydrolysis in the relationships between amino acids and polypeptides. Condensation synthesis joins two monomers to form a polymer, a macromolecule. Water is removed. All chemical reactions within cells require an enzyme. Keep that in mind. For proteins, the monomers are amino acids. The polymer is a polypeptide. When polypeptides are digested into individual amino acids in a process known as hydrolysis, water is required. Now we must dig into some detail. Draw a molecular diagram of a generalized amino acid. Identify amino acids from diagrams showing the structure. Here's a diagram of an amino acid, the monomer of polypeptides, the monomer of proteins. An amino acid has an amino group, NH2, on this side of the molecule, and a carboxyl group, COOH, on this side of the molecule. And very important, there is a variable group adjoining the central carbon. The variable group, or the R group as it's known, is one of 20 possible groups. In other words, there are 20 different amino acids according to the 20 different R groups. Pay close attention. In this diagram, you can see two amino acids. Identify the amino group in each and the carboxyl group in each. Notice the R group. This R group is a simple hydrogen atom, while the R group here is a methyl group, CH3. There are 20 different R groups, thus 20 different amino acids that comprise the proteins of living things. However, and I will say this in words, not in written form, there are some exceptions to the use of 20 amino acids, and only 20 amino acids. There are two other amino acids, 21, 22, selenocysteine and pyrrolysine, that can be found in a few polypeptides in a few organisms. Be aware of this. Here is a single amino acid again, but an important point needs to be made. The R groups can be nonpolar or polar, or charge positive, charge negative. The characteristics of the R groups that make up protein determine the shape of the protein or determine the solubility of the protein. The character of the R groups is very, very important when considering the function of the polypeptide. Keep this in mind as you move forward. In fact, you can see, based on this IB syllabus statement, how important the R groups will be in conversation about protein further down the line. Explain the significance of polar and nonpolar amino acids. So there will be more to the story about the importance of the character of the R groups in determining protein function. Let's look at two other IB syllabus statements that are important. Draw the structure of a generalized dipeptide showing the peptide bond linkage. Outline the role of condensation and hydrolysis in the relationships between amino acids and polypeptides. Hopefully you can see the relationship between these two statements in that the formation of a peptide bond will be a condensation reaction. The breakdown of a peptide bond will be a hydrolysis reaction. In this diagram, notice how two amino acids have been joined by condensation synthesis. The bond, known as a peptide bond, is right here. The R group of the first amino acid is here, the R group of the second amino acid is here, and the entire molecule is known as a dipeptide. The first two monomers, 1 and 2, have come together in the formation of a longer polymer. Notice the NCC backbone. Can you see the pattern as the polymer would grow? Stay tuned. Here is a diagram displaying condensation synthesis and the formation of a polypeptide. Amino acid 1 is joined with a peptide bond to amino acid 2 by condensation synthesis, which removes water. So the role of condensation synthesis and hydrolysis and the relationships between amino acids and polypeptides should be familiar to you at this point, but can you draw a diagram to show the formation of a peptide bond? Here's another image of the formation of a peptide bond with amino acid 1 and amino acid 2 coming together to form a bond here. Notice that this is a process by which condensation synthesis forms the polymer known as a polypeptide. Notice the R group on each amino acid. Notice the placement of the bond between the carbon of the carboxyl group on the first amino acid and the nitrogen of the amino group on the second amino acid. Notice also the removal of water. Here's a polypeptide composed of 4 amino acids. Notice the R groups. Notice the positions of the bonds. And notice the NCC, NCC, NCC backbone. The primary structure of the polypeptide is the number and order of amino acids in the polypeptide. What's interesting, and something you should keep in the back of your mind at this point, is that the sequence of amino acids in a polypeptide is determined by the DNA of an individual. We will look at this relationship carefully later in the course. Remember that I mentioned that the polarity or lack of polarity of R groups was important? Let's take a quick look at R groups once again. In this diagram of an amino acid, we can see the backbone right here. Notice the NCC. This is the R group, and it's nonpolar. In this amino acid on the right, the NCC backbone has been simplified to a square. Keep that in mind. The detail is provided with the R group, which is also nonpolar. Again, in this image of an amino acid, the square represents the NCC backbone. This is the R group. Methionine is also nonpolar, and methionine contains sulfur. Methionine is an essential amino acid, meaning that we cannot synthesize methionine. We must obtain methionine from our food. 12 of the 20 amino acids can be made by the human body, but 8 must come from what you eat. These 8 are called essential. This amino acid, cysteine, with its NCC backbone here, is polar. Cysteine also contains sulfur. Just as a note, you are not expected to recognize which amino acids are polar and which are not, but you need to know that some amino acids are polar, some are nonpolar, and some are charged positive and some are charged negative. Serine and threonine are both polar. Maybe you can see the hydroxyl group in each that would ionize in water. Remember, you're looking at the R group here and here. The NCC backbone has been simplified to the square. Lysine and aspartic acid have R groups that are charged. Lysine is positively charged while aspartic acid is negatively charged. Do you remember what essential means? It means we cannot synthesize the amino acid and we must obtain it in what we eat. Again, here is a dipeptide, 2 amino acids joined by condensation synthesis to form a peptide bond. You can see the NCC backbone and 2 R groups. Remember, R groups can be polar, nonpolar, or charged positive or charged negative. The polypeptide is a polymer composed of a chain of amino acids. The primary structure, determined by the DNA, is the number and order of amino acids in the polypeptide. Here are 3 IB syllabus statements of which you need to gain control. Amino acids can be linked together in any sequence, giving a huge range of possible polypeptides. The amino acid sequence of polypeptides is coded for by genes. This is the DNA. The sequence and number of amino acids in the polypeptide is called the primary structure. Here's a polypeptide, 4 amino acids long. You can count the 4 different R groups here. Remember that amino acids can be linked together in any sequence, giving a huge range of possible polypeptides. Here's a nice diagram displaying condensation synthesis and the formation of the polymer, the polypeptide. We can see the NCC backbone with projecting R groups here. The number and order of amino acids, known as the primary structure, is coded for by DNA in the genes. Here is a polypeptide with 5 amino acids. Keep in mind that the amino acid sequence of a polypeptide is coded for by a gene. The sequence of bases, A, T, C, and G, in the DNA of a gene determine the sequence of amino acids in the polypeptide. There are a huge range of possible polypeptides. If we consider a polypeptide with 100 amino acids, the number of possible sequence is 20 to the 100th power. But only a small proportion of a possible sequences is ever made by living organisms. Only 2 million or so different polypeptides have been identified at this point. Particular sequences of amino acids are made in very large numbers by cells because they have useful properties. Condensation synthesis joins amino acids into polypeptides with the removal of water. Hydrolysis breaks polypeptides into amino acids with the addition of water. Both reactions occur under the influence of enzymes. Now we take our understanding of protein structure to a new level with these IB syllabus statements. The secondary structure is the formation of alpha helices and beta pleated sheets stabilized by hydrogen bonding. Explain the 4 levels of protein structure, indicating the significance of each level. Hydrogen bonding between different amino acids along the polypeptide cause the polypeptide to take on a helical shape. This is called secondary structure. The helical shape is seen here. This is called secondary structure, and it occurs due to hydrogen bonding between different amino acids along the length of the polypeptide. Another look at the secondary structure known as an alpha helix. As well, polypeptides fold and come together under the influence of hydrogen bonds to form the secondary structure known as beta pleated sheets. The next IB syllabus statements are: the tertiary structure is the further folding of the polypeptide stabilized by interactions between R groups. Explain the 4 levels of protein structure, indicating the significance of each level. Folding of the secondary structure, alpha helix and beta sheets, results in what's known as the tertiary structure, the 3rd level structure. In this diagram, you can see the alpha helix and the beta pleated sheets being folded back on itself into the tertiary structure. The tertiary structure is a 3-dimensional conformation of the polypeptide that's determined by the original amino acid sequence, the primary structure. The interaction of specific R groups maintains the tertiary structure. Here you can see the alpha helix folding back on itself, resulting in 3-dimensional structure known as tertiary structure. The 3-dimensional conformation of a polypeptide is determined by the amino acid sequence. The positioning of specific amino acids along the primary structure determines how the polypeptide will fold. And the shape of the polypeptide, the tertiary shape, is critical to its functioning. Remember, the amino acid sequence, the primary structure, determines the 3-dimensional conformation of a polypeptide. The positioning of specific amino acids along the primary structure determines how the polypeptide will fold. The shape of the polypeptide is critical to its functioning. The tertiary structure is the secondary structure folded back on itself, held in position by specific R groups along the polypeptide that interact to form hydrogen bonds or ionic attractions or nonpolar interactions or disulfide bridges, which are covalent. These 4 forces that result from polar, nonpolar, or charged R groups and their specific position along the polypeptide hold the precise shape of the polypeptide. The shape of the polypeptide is critical to its function. This is myoglobin using molecular visualization software. Now you should be able to see the importance or the significance of polar and nonpolar amino acids and their position along the primary structure of a polypeptide is determined by the genes, the DNA in the genes. Here's an antibody using visualization software. Its tertiary structure is held together by interacting R groups that form hydrogen bonds, ionic attractions, nonpolar interactions, and disulfide bridges. So here's a review of the 3 levels of protein structure that I've covered so far, with one additional level, the 4th level structure. Primary structure is the sequence and number of amino acids in the polypeptide. Secondary structure is the alpha helix or beta pleated sheets. The tertiary structure or 3rd level structure is a folding of the alpha helix or beta sheets back on itself. And the 4th level structure, sometimes called quaternary structure, is when more than one polypeptide come together to form the complete protein. Here's a nice diagram to study because it shows the 4 forces that hold the tertiary structure of a polypeptide. You can see the hydrogen bond here formed by 2 polar R groups. You can see hydrophobic interactions here formed by 2 nonpolar R groups. Here is an ionic attraction formed by 2 oppositely charged R groups. And R groups with sulfur will form disulfide bridges, actual covalent bonds. Here are important IB syllabus statements which should appear familiar to you. The quaternary or 4th level structure exists in proteins with more than one polypeptide chain. Explain the 4 levels of protein structure, indicating the significance of each level. A protein may consist of a single polypeptide or more than one polypeptide linked together. Hemoglobin is a protein with 4 polypeptide chains. This is 4th level structure. As well, each polypeptide in hemoglobin has a prosthetic group known as a heme group. The heme group has an iron atom at its center. Hemoglobin transports oxygen in the bloodstream. The forces that hold the 3rd and 4th level structure of polypeptides can be disrupted by heat or changes in pH. This is known as denaturing the protein. And you can see the fully functional tertiary shape of the protein here and the denatured protein here. Again, the denatured protein comes about as a result of a change in temperature or a change in pH. You've had experiences with denatured protein, for example, after you've cooked an egg or after you've used lemon juice to marinate meat or fish. Heat increases the kinetic energy of molecules, thus disrupting the intermolecular forces that would otherwise maintain the tertiary shape of a protein. Changes in the concentration of hydrogen ions also disrupts the intermolecular forces that would otherwise maintain the tertiary shape of a protein. And that brings us to the end of Molecular Biology Part 2, the first installment of proteins. Molecular Biology Part 3 will wrap up our look at proteins.